So thank you so much for having me. And I want to um, say I'm excited to be part of this group. And yeah, I hope that I can tell you a little bit about this newer project that I've been working on in a rap model of vulnerability to postpartum mood disorders. Um, so, most important is that postpartum depression is a serious concern. So, about 86% of American women will eventually become mothers. And of these, about percent of women experience what we call the baby blues, which is just this kind of shift in mood and increase in anxiety that occurs in the weeks postpartum. Fortunately, many women get through that period as hormones and other factors regulate. But unfortunately, 30% of about 30% of these women will continue to experience more intense and debilitating symptoms that we call postpartum depression or generalized postpartum mood anxiety disorder. Because anxiety is, of course, another facet in this disorder, not to mention postpartum psychosis, but I don't go there yet. But so the symptoms associated with postpartum depression really are a depressed mood, this sadness, hopelessness, significant sleep disturbances beyond just that of having an infant in the first 9 months of life, fatigue, difficulty concentrating, recurrent thoughts of suicide, and severely diminished interest, pleasure in most activities, which we call anhedonia as a symptom. And that, most importantly, translates to spending time with one's new baby. About 5 to 14%, and so the numbers are just still, what you'll see from this is research in postpartum depression is really in its infancy. But these numbers range between 5 to 14% will contemplate self-harm. And unfortunately, postpartum depression is one of the major risks of childbearing, and suicide is one of the leading causes of maternal death in the United States. So we hypothesize that this indicates that doctors are unlikely to effectively identify, diagnose, or treat postpartum depression within the time frame that it emerges. And that's not really surprising given what we know. I mean, we have a baby, you're at the hospital for 2 days, you go home, and your only maybe follow up is a 6 month follow up with your OB, and that's about it. So, it actually tends to fall into the pediatrician to sometimes ask some of these questions. And yet, it's interesting because the American Psychiatric Association and the World Health Organization do not consider postpartum depression a distinct mental health disorder. They consider it a subtype of major depressive disorder with its onset occurring at or around parturition, um, around the time of birth. They used to say in the last diagnostic statistical manual, they used to say that it was only postpartum. Now in the DSM-5, they'll at least acknowledge the fact that there can be prepartum or peripartum onset for a lot of these symptoms. That said, there is currently insufficient um, evidence to differentiate postpartum depression as a separate disorder because actually when you look at like the research done in depression, less than 0.2% has even considered the postpartum period. So if you just do a PubMed search on depression research models, very few have considered actual females, let alone postpartum period. So that's what we're interested in. And we're really interested in it because the postpartum body is a unique time. There's a lot of brain development and plasticity that occurs in the mother's brain during this time. We know this from both rodent and human studies. This is a very unique endocrine state, right? The hormones of pregnancy, you'll never see those levels at any other time except during pregnancy and that rapid change that occurs postpartum. In addition, there's a lot of changes that are associated with the immune system and inflammation associated with parturition. So I'll go through a little of these each independently and why. And these are really factors that my lab has long been interested in, is just understanding how the body and brain act to influence brain function and behavior. So we know that pregnancy and maternal experience can fundamentally change the brain. So in women, there is MRI and fMRI evidence for hormone-dependent changes. There is a postpartum gray matter volume is consistently lower in regions associated with social cognition. Mothers across various cultures show higher midbrain and striatal activation in response to baby's cries compared to other sounds. The extent to which a mother's brain responds to her own baby's cries actually varies with the type of delivery method. So, whether it was a vaginal birth versus cesarean, and the method of feeding, whether you're giving bottle versus breast. And so, this also influences the maternal brain and how it responds to cries in an fMRI study, suggesting that the actual process of birth and that type of maternal care influences the brain. In individuals with postpartum depression, the maternal fMRI response to her own baby's cries and her own baby's face is lowered. So it suggests that those changes that are occurring in the brain are not happening to the same degree in women with postpartum depression. And a lot of my work as actually an undergraduate student now, 20 plus. Oh, do you guys hear me? Okay. I thought I heard something. But can you guys hear me okay?
Yes,
We
Jackie,
can hear you. Yep.
Yes. Okay, I thought I heard somebody trying to say something. Anyway, so this is what I actually did when I was an undergraduate student, was just understanding how pregnancy and maternal experience changed the brain. And I used a rodent model back then. So in rodents, this brain development, these changes that occur in the postpartum period are necessary for the expression of appropriate maternal behavior. There's some kind of plasticity that's occurring. So in particular, the medial preoptic area, which is a structure just outside the hypothalamus, receives changes in estrogens and progestins as pregnancy continues, and oxygenergic input. And all of that is necessary to induce appropriate maternal behaviors. In fact, a female rat will not show maternal behavior until she has received these appropriate changes in maternal hormones. And the preoptic area is interesting because it seems to be a central hub that drives changes in behavior. It reduces levels of activation in the amygdala, but it also drives mesolimbic dopamine activity. So that if you inhibit the medial preoptic area, if you inhibit the ventral tegmental area, which is the structure that produces those reward projections into the brain and nucleus accumbens, or if you antagonize dopamine receptors and the nucleus accumbens in particular, and that's work I did as an undergrad, it all inhibits maternal behavior, which suggests that engaging in maternal behavior and care is a reward behavior. But this plasticity is stress sensitive. So that's another thing to consider, is that if stress is involved during this process, we can disrupt a lot of this plasticity as well. So as I was starting to allude to, the hormonal changes of pregnancy and delivery are important for maternal behavior. If a virgin female rat is given pups that she's never seen before, she'll avoid them, she'll attack them. But if we were to follow treatment paradigms, and something I also did as an undergraduate, with a paradigm that mimics those hormones of pregnancy, an increase in progesterone followed by a spike in estrogens, we can induce maternal behavior in those females. So we know that this is essential to eliciting changes in the brain. In addition, factors that affect the hormonal state, either delivery method or stress that occurs at the time of gestation, can also affect the maternal brain. In clinical postpartum depression patients, there are differences in the ratio of estradiol to progesterone that have been predictive, like say in the second and third trimester, that actually predict the onset of postpartum depressive symptoms, suggesting that there's a priming effect. And stress axis dysfunction and higher levels of cortisol have been observed in postpartum depression patients. Women with postpartum depression exhibit depression anxiety symptoms. They also show differences in the levels of circulating pregnenolone. Now, that's a hormone that's a metabolite of progesterone. And some of the most recent drug treatments for postpartum depression target pregnenolone. In addition, pregnancy alters immune function, and this is something that's really not been explored at all. Um, Females are generally immunosuppressed, what we would say, during pregnancy. So during pregnancy, you can see an attenuation of a lot of autoimmune disorders, and this will decrease, but then right after birth, they increase, they'll have a rebound effect for a number of different disorders. In this case, there's this particular paper showing Hashimoto's, thyroidosis, and Graves' disease, but the same can be said for multiple sclerosis and lupus. So we think that this is due to an immune rebound that occurs postpartum, just the way that there's an endocrine decrease postpartum. Yet none of these things have been examined in the context of postpartum depression, but we think it's important because inflammation has been associated with depression in general. And patients with treatment resistant major depressive disorder do often show higher levels of inflammatory cytokines. And it's been proposed that cytokines could serve as biomarkers for depression and predict treatment outcomes. So, what we're really interested in understanding as our research question is, one, do rats show differences in postpartum mood or anxiety, just at baseline? And then can we use that to examine risk factors or some of these inflammatory or endocrine mechanisms that may be associated with postpartum depression? So what we did first, the symptom of anhedonia using a sucrose preference test. In this case, our preference paradigm is to place two bottles, one of which is just plain water, tap water, the other of which is a 1% sucrose solution in the cone cage. And after 24 hours, we weigh the bottles and place them back in the cage with the sides reversed. And after 48 hours, we weigh the bottles again, and then the test ends. The goal was to test this before breeding, so at baseline, and then again immediately postpartum on and again on postpartum day 7 through 9. With this data, which is the amount of sucrose solution that's consumed as a total of the amount of fluid consumed, and obviously the percent, then multiplying it by 100. So this is what the data looks like at first. Okay, so if we just give female rats 1% sucrose solution in their water bottle, this is what the data looks like. And as you can see, a lot of animals love the sucrose solution. So will be the sucrose solution. And so we kind of exclude those from subsequent analysis, but we're starting with these females that clearly love sucrose. Now, what does it look like postpartum? And this is where we were really surprised about the data, because we actually found a lot of variability in the data. There was a group that clearly continued to love the sucrose solution, and then there was a group of females that show a significant decrease in sucrose preference. And then there was some, and we actually excluded these. And what we did from this is we created these kind of two groups: females that were resilient to this postpartum anhedonia, and a group that was susceptible to this.
Okay.
But
Hey,
a
Jackie, it's
subset of
Rob.
them.
Can I
Yes,
interrupt for 1
Rob.
second? I have a request. Could you turn off your your camera? Because there's a little bit of like breakup in the audio part. So maybe if you turn off your camera.
Yeah, I might have to okay. I might have to come back here, turn off the camera, and come back here. See that?
Okay,
We'll
thanks.
see if it's better. Okay. And so we see the again, this susceptible and resilient phenotype persisting into postnatal days 7 and 9. I kind of broke this down by a table because what you can see is that when we start with the total number of rats, right, there's some rats that aren't interested in sucrose. It could be actually that they didn't figure it out, right? That's another issue is we give them 48 hours and maybe some of the rats don't figure it out that hey, there's a better solution over here. But nonetheless, we exclude those and then we can see this separation in the susceptible versus resilient dams. Most importantly, is that we looked closely at the number of dams that switched from the susceptible to the resilient condition, and really it was just one. And only one female switched from the resilient to the susceptible condition. So there was a persistent phenotype even until the postnatal day 7 and 9 days. So the next thing we did was take these animals and do a simple measure of maternal motivation. This is a pup retrieval test where you take the litter from, say, if she keeps her litter back in this little nest, we would take a few pups and divide them across these other areas. And then we time her and say, how long does it take you to retrieve your pups? Because based on their calls, she should pretty much go right away and collect her pups. And we see this significant difference where the susceptible dams are slower to retrieve their pups back to the nest, suggesting there may be a difference in maternal motivation. And we have other ways we plan on testing this in the future. But in addition, we were surprised it wasn't significant with this end, but that there was a trending decrease in the size of the pups. So they weighed slightly less when their moms were susceptible. But that may not be surprising because we actually did a comprehensive assessment of other types of maternal behaviors. And we saw significant differences, particularly early in the postpartum period, in the amount of pup licking, which is what I've shown here. But we also saw it in certain aspects of nursing behaviors, suggesting that maternal care isn't as robust in the females that are susceptible than the resilient females. We also tested anxiety. So, same females. Are you susceptible or resilient? And now we put them in the elevated plus maze and look at the time and the number of entries into the open arms, which should be the more anxiety producing areas. And do they stay out of these areas if they're anxious? And in this case, we do see a significant difference in the elevated plus maze with the susceptible dams, more likely to show that anxious behavior in the elevated plus maze. And this was performed on postnatal day 3, which is actually why back here we had to take a break on postnatal day 3 to do some other behaviors. So yes. So we're seeing this interesting phenotype. And I'm kind of wrapping up. Um, so we've got this divide between just at natural baseline, there's a number of females that are susceptible to, first of all, postpartum anhedonia. There's others that continue to enjoy engaging in the sucrose solution. These same females that are susceptible are also likely to be more anxious. They are showing decreased levels of maternal care, especially immediately postpartum. Of course, maternal care behaviors tend to decrease across time until they wean. And the pup weights were slightly lower. Now, what I didn't show you is that we've also looked into the brains. And in the brain, we see this consistent increase in BDNF. It's a huge increase in BDNF that's actually in the prefrontal cortex. That occurs in the first 2 days postpartum. But in these females that were susceptible to this phenotype here, we see a decrease in BDNF. We don't see the increase, and we also see an increase in the inflammatory molecule IL-6. So some of our future experiments are going to be testing some of the mechanisms as they relate to this behavioral phenotype. One thing I also will say is that we measured stress hormones and um, and estradiol levels at least. And we saw altered profiles. So we're not sure how to understand that, but we know now that at least the endocrine profile was significantly different. It's just shifted in a way that seems to be different from the normal paradigm. Because actually, when we were testing how to measure fecal steroid hormone levels, which is so fun to collect poop and measure steroid hormones, we did it from just standard pregnant females, males, estrus females to see if we could validate the model. And we saw these very altered levels of hormones. So our next steps, I've been writing a grant. We're going to try to take this into the brain a little bit more. So we've been looking at neural connectivity using functional MRI. We know that functional connectivity increases in the default mode network of postpartum women, and that when these changes don't occur, there's deficits or variation in this default mode network. They are not what they should be, that actually predict deficits in postpartum behavior. So what's nice is with our model is we can look at baseline, postpartum, and late postpartum to see whether we see similar changes in functional connectivity or any changes in structure that may be different between resilient and susceptible dams to see what might predict or correlate with the onset of the behavioral phenotypes. We're also looking at these changes in other molecular biomarkers in the resilient and female dams, susceptible dams, and comparing peripheral biomarkers so that we can continuously compare brain and peripheral cytokine expression. In our second aim of our experiments, we're looking to specifically modulate that IL-6 that was upregulated in the susceptible dams. And we think that this might be a mechanism by which we're increasing that susceptibility to postpartum depression and decreasing that maybe some of that plasticity that is necessary for coping with this change in their lives at this time. And I know I'm making them sound like new moms, but they are new moms. So we'll be testing this by infusing an IL-6 receptor antibody into the prefrontal cortex to reverse and see if we can reverse that anhedonia. At the same time, we'll be doing some of our functional MRI, which we have here, of the default mode network to see if we can change some of that as well as alter peripheral biomarkers. And then on the flip side, can infusion with the recombinant IL-6. Now, we've had good luck with the IL-6 receptor antibody because this is actually a pharmacological drug that's used to treat a number of disorders. But in the flip side, we're going to see if we can use the recombinant IL-6 to induce susceptibility to postpartum anhedonia and really see whether it's sufficient to induce this anhedonia phenotype in the resilient dams, and then again, examine the brain and biomarkers. So those are some of our next steps, and our grants keep getting pretty good scores, just not good enough. So, so yeah, that's my story, and I'll just thank everybody and some of the past fundings that has gotten me to where we are in this project.